Hello, everyone, and welcome to the Parkinson's Foundation program, New Frontiers in Research and Care. I'm Sarah Maldonado, Community Program Manager for the Parkinson's Foundation in Arizona, and I'm excited to be here with you today to share this educational program. I'd love to take a second and hear from our audience. Let us know in the chat, where are you joining us from today? I know from chatting with a few of you before the event, we had people from all over Arizona and even a few outside of the state. Give you guys a couple seconds to share where you're joining us from. All right, thank you all so much for sharing. We are so glad you guys are joining us today. We hope you know that you are not alone. And as you can see from the chat, your Parkinson's community is here with you from near and far. For those of you who are new to the Parkinson's Foundation, welcome. We are the nation's leading community for people with Parkinson's disease, the people who love them, and all of those who are working to end the disease. With our presence in communities across the country and the globe, we believe in the promise of a cure and a better life for those impacted by PD. The urgency of our mission really translates into what we do. To achieve our mission, we pursue three goals: ensuring better care for everyone with Parkinson's, advancing research towards a cure, and educating and empowering the Parkinson's community. We provide free resources, including our website at parkinson.org, educational book series, webinars, podcasts, a hospital safety kit called Aware and Care, and our newly diagnosed materials. And of course, our toll-free helpline at 1-800-4PD-INFO. On the research front, we invest more than $10 million annually to study Parkinson's, what causes it, how to treat it, and ultimately how to cure it. We'll be covering PD generation in more detail and sharing how you can participate during today's program, so stay tuned for more information. Speaking of research, let's jump right into our presentation. I would like to introduce our first speaker, Dr. Holly Shill. Dr. Shill is the director of Muhammad Ali Parkinson Center and Movement Disorder Program at Barrow Neurological Institute, Dignity Health, St. Joseph's Hospital Medical Center in Phoenix, Arizona. She's a professor in the Department of Neurology at University of Arizona, College of Medicine, Phoenix, and she is board certified by the American Board of Psychiatry and Neurology. Dr. Shill received her medical degree from University of Arizona, College of Medicine. She completed her internship at St. Joseph's and Medical Center in Internal Medicine and completed a residency in neurology and a fellowship in movement. Oh. One second, looks like I lost my slide. Okay, she then served as a clinical fellow at the National Institutes of Health studying movement disorders. She has received numerous awards over the years, including becoming a National Merit Scholar and being invited into the Alpha Omega Alpha Medical Society. On that note, Dr. Shill, I'll turn it over to you and you can go ahead and share your slides.
All right, is everyone seeing my slides okay? Can I get a quick chat to?
Yep, it looks perfect, Dr. Shill.
Okay, good, wonderful, thank you. So thank you everyone for joining us today. So I'm delighted to be here. I'm visiting you from my office here in downtown Phoenix at Barrow Neurological Institute. And today I'm going to talk to you a little bit about really the importance of research in Parkinson's disease, you know, the importance of participating, what research has told us. And I'm going to share things for about 20 minutes or so for my presentation this morning. So or this afternoon, I guess, for some of you. So our learning objectives today: to understand the important role research plays in the search for better treatments for Parkinson's disease, exploring ways research has shaped current treatment and care strategies for people with Parkinson's, providing information on current Parkinson's Foundation research initiatives, and identifying new treatment and care strategies that can help improve day-to-day functioning for the management of Parkinson's symptoms. So I first thought I would talk a little bit about the types of research that we do. This is just kind of in medicine in general. So you get a bit of an idea, a bit of an overview before I jump specifically into Parkinson's disease. So the first type that we think about is epidemiologic type research. So, and this is where you just take tons of data from people, lots of clinical data, maybe biological data, that type of thing. You kind of mish it all together and you throw it at the wall and you see what sticks. So an example of that would be something like the studies that we've done in isradipine. there were some interesting studies in Parkinson's saying that people on this specific type of calcium channel blocker were actually at lower risk for developing Parkinson's disease. And so this just led to some interest in whether this drug might slow progression of Parkinson's, and eventually to some studies on it. So it turned out, unfortunately, that the drug didn't work to slow progression of Parkinson's. But we learned a lot about how this epidemiologic evidence then kind of translated into Parkinson's research. So another type of study would be observational type studies. And this is just where you simply follow people along and try to learn a little bit about more how Parkinson's progresses. So a good example of that is the PPMI study, the Parkinson's Progression Markers Initiative. So then we move into preclinical studies, and these would be things like animal studies, you know, maybe specific genetic mutations in animals, and looking at what their behavior looks like and what their biology looks like. And then probably more relevant for everyone here is studies in what's called disease modification, trying to slow progression of Parkinson's, as well as specific treatments for the symptoms of Parkinson's, whether you're looking at mobility or whether you're looking at you know, depression or non-motor symptoms of Parkinson's. And then finally, there'd be studies that would be kind of social science type studies. You know, what's the best, you know, maybe way to deliver care in a clinic, for instance. So, you know, how do you study telehealth in Parkinson's? So those are the types of research that we're going to be talking about. And I'm going to kind of go over that all using a very specific example in Parkinson's. And so to start off this little journey, if you will, we're going to just talk about what we know as the kind of biochemical basis of Parkinson's, or the main kind of biochemical or pathologic abnormality in Parkinson's. And we all kind of know this, but just to go over it, it's this concept of reduction in dopamine in the brain, and particularly in an area of the brain called the substantia nigra. So that's where these little dopamine neurons live in the brain. And then they project throughout the brain to help us with our movement, to help us with our thinking, that type of thing. So, and I think we all know that there has to be enough of a loss of that dopamine before what we call the cardinal signs of Parkinson's then are manifest. So that's the one feature, is this biochemical abnormality. And then the other thing is sort of the pathology. So what you see in the brain. And so these slides, the two slides of the brain down on the left-hand side of your screen demonstrate what are called Lewy bodies in the brain. So these are these little accumulations of protein in the brain. And it's been known for, you know, over 100 years that these Lewy bodies exist in the brains of people with Parkinson's, but for a long time, it was really not understood, you know, what it was all about, and, you know, what was actually in those Lewy bodies and that type of thing. So, so to go on this journey of the Lewy body and alpha-synuclein. So we're going to talk about the story of alpha-synuclein to just kind of get an understanding of how we came to where we are today, you know, so 20, 30 years of studying these Lewy bodies in the brain and the alpha-synuclein, and how is that informing us? So, so the story starts off in the clinic. There was a neurologist by the name of Dr. Larry Golby, who I've met before as a longtime neurologist and working in New Jersey in the spring of 1986. And he happened to have a patient in his clinic who happened to be a little bit of a younger onset Parkinson's patient. So he'd had Parkinson's for 10 years, but it started at a younger age. So, and he didn't think, you know, too much about it. You know, younger onset was a little bit atypical, but other than that, didn't think too much of it. So anyway, the patient ended up passing away, and afterwards, the brother of the patient came to Dr. Golby and said, hey, I think I have Parkinson's too. And sure enough, the brother was diagnosed with Parkinson's, and both of them at a relatively young age. So, and so this got Dr. Golby thinking, and it kind of turned out that both of these patients were Italian and from a small city, a small town in Italy called Contursi. So, this is kind of interesting, and he didn't think too much of it. At the time, there wasn't really a lot known about genetics of Parkinson's. So then a few months later, Dr. Golby happened to run into another patient in his clinic who also had Parkinson and also was Italian, and also from the same small town. So, and this really got Dr. Golby thinking. He's saying, this is kind of unusual to have all of these younger onset Parkinson's patients, all that happened to be from the same town. And so he, working with his group at his institute, decided to actually meet with some of the people in Italy, to actually go to Italy and start to look at these people that are coming from this area and whether there is anything to this sort of heredity of Parkinson's. So, and this led Dr. Golby and his colleagues, I'm just putting the initial publication, to put together this large kindred of people. So kindred meaning a huge family, lots of generations of people living with Parkinson, or living and deceased in multiple generations of folks with Parkinson's, who all seem to come from this very same couple over 500 years ago, who kind of were the original people of the family, of which this gene seemed to be passed down. So, and so it's an interesting story of him going off to Italy, and they actually were looking through like baptism records and marriage records. And fortunately, they actually kept all of these records. And it turned out the doctor who worked in this small town had been working there for a long time as well, and so knew a lot of these family members. And they were actually able to pull a lot of these family members together, get blood from them, send them back to the United States, into New Jersey, to start the genetic analysis. So, so you get all of these family members, all of these people, you know, it was, I think, about 60 people altogether that ended up having Parkinson's disease and submitting samples for this study. And that led to our first sort of understanding that there may be genetic components to Parkinson's. So next part of the study. So in describing these patients, the younger onset, they seem to have a more rapid course of Parkinson's. But very interestingly, these patients seem to have very typical Lewy bodies. So a couple of the patients had come to autopsy, and they were able to look at them under the microscope and see that they had these very typical Lewy bodies. So, and because of the nature of the heredity, where they were seeing it in generation to generation, they postulated that a single gene was responsible. So they started hunting for the gene. So they had blood on a number of these blood samples on a number of these patients, and started really hunting for the gene. And it took them years and years before they finally were almost ready to give up. They'd kind of put it to rest, but then decided to collaborate with a group at the National Institutes of Health. So, and very rapidly, with just kind of improvement in genetic technologies and all, they were able to find actually the part of the genetic chromosome where this gene lives. So, and the way they do that is they kind of look at the people who are affected and then compare their blood to the people, compare their genetics to the people who are not affected. And they say, okay, what's different in these groups of people? And this allows them to sort of march down the gene and eventually do what's called linkage. You link it to a certain section of the gene that seems to be distinct between the two groups of both affected patients and then unaffected, their family members and things that don't carry the gene. So, and so for a while they knew the linkage, but they actually weren't sure of the gene. So, and then with kind of even more sophisticated genetic analysis, they were actually able to pin down the very region of the gene that was responsible, and then went to a library and found out, hey, this gene looks like it's this gene called SNCA, which is the gene that codes for this protein called alpha-synuclein. And so that was the publication now back in 1997 that linked alpha-synuclein protein now to this genetic form of Parkinson's. So, and for a while people said, oh, isn't that interesting? This small family of Parkinson's, not really seen. I mean, they did other families with Parkinson's, et cetera, and not really finding anything there. So it seemed like it was a fairly rare cause of Parkinson's. So, and to this date, it actually still is a fairly rare cause of Parkinson's. But what's very, very interesting is that they found that this alpha-synuclein protein is actually in the Lewy bodies. So even in people who do not have this genetic form of Parkinson's, the alpha-synuclein lives in the Lewy bodies. So, and this was just almost discovered accidentally. It was an Alzheimer's researcher who was just making sort of antibodies to this protein, and happened to be looking at brains of people with Parkinson's and realized that this alpha-synuclein was what was living in the Lewy bodies. So, so that was a very, very interesting finding. And there was this sort of aha moment that maybe this gene is relevant for this gene as well as this protein is relevant for more than just kind of this small family of hereditary Parkinson's. So the next thing that came out of this, and this was a little bit later on, was this large Iowa family. So So they didn't carry the mutation for the alpha-synuclein protein, but what they actually had was a triplication of the protein. So, so instead of a break or a mutation, an abnormality of the genetic material, there was actually too much of the genetic material. And so instead of having less of the protein and having a kind of a disrupted form of it, these people just simply made more of that protein. And they learned that that was actually sufficient to cause Parkinson's in this family. So, so what we learned that was more of alpha-synuclein was not good. So it's not good to have a mutated form, but it's also not good to have too much of it. So, and so this really led to this idea of looking at alpha-synuclein specifically as relevant to Parkinson's. So another thing that had to happen before we could do extensive research was to find the best way to find alpha-synuclein in both the brain as well as actually in the body. And so this was parts of studies that happened here in Arizona at the Brain and Body Donation Program out in Sun City, Arizona. So they actually looked at a variety of different ways to measure alpha-synuclein, and this was research done with the Michael J. Fox Foundation. Looked at different ways to find alpha-synuclein in the brain, a bunch of different ways to kind of assay it. And then they came up with the winner down at the bottom, which was this, what's called LB509 with a specific type of processing. So this ended up being the winner. And so now this has kind of been the gold standard if you want to try to kind of find alpha-synuclein in the brain as well as to find it in the body. And so this next led us, now that we've got this good way to find it, to look for it in the brain and the body of people with Parkinson's, now we can say, okay, how do we actually, can this help us stage Parkinson's? So we know that this protein's abnormal, we know that it accumulates, we know that too much of it is a problem. So the next step is to say, okay, how does it accumulate? So how does it start? How does it accumulate? And this is led to some studies looking at pathology in Parkinson's to say that we can actually stage Parkinson's pathologically now. So we know the different areas of the brain, and some of you know that that we now know that Parkinson's seems to start in the sense of smell. So you can actually find this funny alpha-synuclein protein in the olfactory bulb in the brain, and that actually may be one of the earliest places that it is found. So there's some thought that that's kind of where Parkinson's starts. So and then it's actually not until it gets into the substantia nigra then that you start to see the dopamine deficiency and see the kind of typical motor features, the tremors, the slowness of movement. So the other thing that we have been able to do with being able to stain now to look at the alpha-synuclein in the body is to look for, well, is there Parkinson's outside the brain? So we think of Parkinson's as a brain disorder, and certainly that it is, but can we look at it outside the brain? So, and we've, for some of you who live in Arizona, you know that we've been doing whole body donation. So for people who participate in our brain and body donation program, we do whole body donation, and that's allowed us to look for kind of, is there a place in the body maybe where Parkinson's might live? And there's a couple of key places where it lives. So the submandibular gland, these little salivary glands that live underneath the jaw, the lower part of the esophagus, and then a place called the sympathetic ganglion in the brain. So, and this has kind of led us to think about, is there a place now to potentially, biopsy to find a site for Parkinson's? I'll talk a little bit more of that in a minute here. So, rather than biopsy the brain to go look for this alpha-synuclein, can we look for it outside the brain? So, and I'll talk about that in just a second. So, with the staging studies, we've also learned that we can clinically stage people. So for instance, we know that people with reduced sense of smell and abnormalities in their dopamine system as measured by what's called DAT imaging are higher risk for converting over into typical Parkinson's disease. So we know that the people that have the REM sleep disorder, so have the very vivid dreams and act out their dreams, are more likely to go on and develop the classic signs of Parkinson's disease over time. So, and this has really all come out of research being able to show that these are areas of the brain that are affected with this alpha-synuclein or with the Lewy bodies even before the substantia nigra, or the dopamine system, is affected. So this has helped us understand the clinical progression of Parkinson's, not just what's happening in the brain, but also what we're seeing in the clinic as part of Parkinson's progression. So, and then I told you we'd talk quickly about diagnosis. So can you measure alpha-synuclein in the body? So we've talked about a couple places where it's very high levels of the alpha-synuclein in the body. And this has led us to, can we biopsy these sites to actually help us make a diagnosis of Parkinson's, potentially even very early on where the symptoms are very mild or maybe more difficult to pick up. So we've been doing studies in Arizona here on biopsying of the submandibular gland and shown that it's about 80% accurate in picking up Parkinson's. So we biopsy, and ear, nose, and throat goes in and biopsies this little area, submandibular gland, and sends it off to the lab for analysis. And it's about 80% accurate. So it's not 100%. It's sometimes hard to actually pick up the protein, even though it's in high concentrations there. It's sometimes hard to pick it up. So there was a study called the S4 study where they looked at a variety of sites to study to try to biopsy for Parkinson's. And one of the things that has kind of come to the forefront now is actually skin. So, and some of you may have heard of this, but it is possible to measure alpha-synuclein in the skin. So when we originally did our studies, it wasn't that. When we did the large staging studies, we really didn't find high concentrations in the skin. So, but over time, the technologies are improving significantly, such that we're getting really high, as high as 98% accuracy in biopsying the skin. So those areas of that area of research is still research. It's not quite commercially available, but I think it is up and coming as a promising area for research in Parkinson's. So. And the last to kind of finish with you before I finish and turn it back over is on how does this alpha-synuclein business translate into treatment? So you've heard about, it helps us with progression, it helps us potentially with diagnosis. So, but can it actually help us with treatment? So, and so I think there's a couple of things where I think we're learning that it can potentially help with treatment. So first is just trying to understand how and why does alpha-synuclein accumulate? What is that trigger that sets it off in the first place? So is it some, outside trigger that sets it off, something, a virus, something like that may set it off, something in the environment that may initially trigger it? And then what is the process once it starts to accumulate? What's that process that allows it to kind of propagate? So understanding that will actually help us understand how better to slow progression of Parkinson's. So, and then another area of research is, okay, well, what if it's already started and you start having this alpha-synuclein in the brain? Does clearing it out help? So, and some of you may be familiar with this area of research, which is these monoclonal antibodies now. So you get an antibody that actually helps to sort of clear out the alpha-synuclein in the brain. And those are studies that are underway right now and just starting to report. I think there's some evidence that maybe it's not as great as we thought it was, but I think it's still very much an area of research that we're going to learn more about in the next coming years. So, so this whole kind of story about how the initial recognizing the patient in the clinic as potentially having something genetic that is contributing to their Parkinson's, all the way down through, maybe 20 years now of 20 or even 30 years now, I guess, of study, and now leading to treatments that may actually slow progression and halt progression of Parkinson's. So, so I'll finish my story of alpha-synuclein and just leave you kind of with a little bit of advice here. So the first thing, and you're probably already on this if you're participating in today's program, but to get on the mailing list for clinical trials, basic science research, participate. So learn what's going on, become active. The Fox Trial Finder, the Parkinson's Foundation, the NIH repository, which is clinicaltrials.gov. I looked just 2 days ago and there were 2,828 studies going on in Parkinson's disease. So, so keeping up with what's going on and kind of stirring your interest in it, I think helps at least in me keeping motivated and on the search for the cure here for Parkinson's. So, and then get educated on the medications for Parkinson's disease. Understand the research behind them. Ask your doctor, why are you picking this drug for me? What's the evidence that says that it works? So, and hopefully they are able to tell you about that. So understand what, why, when we use certain drugs the way we do, that type of thing. So exercise, high intensity and often. That is the one thing that is out there that is most likely to slow progression of Parkinson's at this point in time. And so lots of research behind that. So I encourage you to engage in that. So talking with your supplements with your doctor. So making sure your doctor's aware about what supplements and things you're taking. So just because you read about it on the internet doesn't mean that it's fact and that it works. So, and that's my last one is last little point here is be careful about charlatans. So I've learned that if I just Google Parkinson's, I all of a sudden start getting ads about, this is going to cure Parkinson's, that type of thing. So be real careful about what you're reading, whether you're hearing it from a friend or reading it on the internet and that type of thing. So make sure you're kind of vetting these things before you're thinking about it. Make sure before you're using them, make sure you understand the science behind what you're putting into your bodies. I think are all good advice. So, and with that, we'll finish up here. So let me go ahead and stop sharing. And I thank you for your attention.
Great. Thank you, Dr. Shill, for that wonderful presentation. If anyone has any questions based on the information that you just heard, feel free to leave a question by clicking on the Q&A icon at the bottom of your screen. We'll get to as many questions as we can at the end of the presentation. At this time, I would like to introduce our second speaker, Dr. Anna Naito. Anna serves as the Associate Vice President of Research Programs at the Parkinson's Foundation, where she manages a portfolio of the Foundation's flagship clinical research studies, including PD generation and the Parkinson's Outcome Project. Dr. Naito brings extensive experience in PD through her previous role at the Michael J. Fox Foundation, where she served as scientific lead for globally recognized PD studies and managed portfolios that leverage global research experts and industry collaborations. Dr. Naito completed her graduate work at the University of Southern California, where she earned a PhD in molecular pharmacology and an MS degree in regulatory science. Her early research focused on drug targets for neurological disorders. On that note, Anna, I will turn it over to you and you can go ahead and share your screen.
Thank you, Sarah, and thank you, Dr. Shill, for a really wonderful presentation. I will share my screen now. Okay, so every person's journey with Parkinson's is a unique one. So not every person has the same symptoms. They have a different age of onset depending on that type of person and their family history. So when we think about drug development for Parkinson's disease, it's very difficult to come up with a treatment that's going to help all people with Parkinson's. So the most logical place for us to start when we think about better drug treatments for Parkinson's is to really look at genetics, because genetics is really what distinguishes each of us individually from one another. So it can serve as a key to understanding a little bit more about the biology of Parkinson's disease, and we may be able to use genetics to help us diagnose and treat Parkinson's. So our goal here at the Parkinson's Foundation is to provide genetic testing so that we can gather the fingerprint of every person with Parkinson's disease. And when we overlay those fingerprints together, we will be able to see different things that are dysregulated, the pathways that are broken. And we have to know that we can't fix things if we don't know what's broken. So this is really the most logical place to start off, is to look at the genetics of every person with Parkinson's. So I just want to give a very quick example of how genetics has led to the discovery of breakthrough therapies. And I'm going to use Lipitor as one of our examples because it was really genetics, the discovery of a specific gene that controls cholesterol. That discovery then led to the discovery of Lipitor, which lowers cholesterol levels. And we know that Lipitor can help all sorts of people. So it doesn't have to be a person who carries that genetic mutation that links back to the biology. But now what we know is that Lipitor is ubiquitously used. And what we want to do today when it comes to Parkinson's is to be able to get there. we need to start with understanding the different genes that control the disease, and then we can start to build drugs around that so that we can fix what's broken. So we are at a very interesting and exciting time when it comes to Parkinson's research and drug development, because there are so many companies right now that are on the market starting phase 2 and phase 3 clinical trials that are targeting Parkinson's genetic mutations. And so what we hope to do is to be able to help accelerate this research, accelerate these clinical trials so that we can get better treatments into the hands of people with Parkinson's today. And ultimately, what we want to do with this information is to help empower people with Parkinson's disease as well as their clinicians to help make better decisions on their clinical care. So this is just a rationale for the PD Generation initiative that the Parkinson's Foundation launched. Obviously, our end goal is to be able to help find a cure for the disease, but in the immediate term, what can we do? We have to identify what needs to be fixed. And so what we could do now today is to collect genetic data to help advance research forward. And what I want to stress to this group today is that every person can make that contribution towards research, because every person matters. If we don't get better information for genetic information with people with Parkinson's, we'll never be able to resolve the biology of the disease. And so this is what spurred the Parkinson's Foundation to launch PD Generation. This is a study for adults who are 18 and over with a confirmed diagnosis of Parkinson's disease. And this is a service up to 15,000 people in the United States for people with Parkinson's. And what it is offering genetic testing and genetic counseling at no cost to people. And we are offering this for the first time bilingually in both English and Spanish. And we are so thrilled to be able to share that Barrow Neurological Institute under Dr. Shill's leadership will be able to provide bilingual services to offer both genetic testing and genetic counseling in English or in Spanish. I want to emphasize here genetic counseling. What is that and why is it important? Genetic counseling is a very specified field where these are counselors who are highly trained in the field of genetics, but more importantly, what we wanted to do was find genetic counselors who have a specialization in Parkinson's. There are different genetic counselors for cancer, heart disease, pediatrics. And so what we want to do is to be able to provide a standard of care for genetic counseling. And so this is the first study that's offering genetic testing in conjunction with genetic counseling so that we can really help educate people to read their test results and be able to ask those important questions about implications to family members and loved ones. We're also looking to bank the residual DNA for future research. And this is also very important because we're not just limited to the information that we have today. Through this initiative, we'll be able to hopefully find and discover novel genes that are linked to Parkinson's disease. And so by banking that DNA, we can actually pull that extra DNA off the shelf when we have new discoveries and not have to have you come back in to give us a new sample, but really just pull it off the shelf so that we can update you with new test results. So the test is actually looking at 7 key Parkinson's related genes that are in this green box to the side. And I will also emphasize that we're looking at 7 genes because these are the definitive clinically relevant genes for Parkinson's disease. Many of you might ask us, why didn't you include other neurological conditions? And what we did when we initiated this PD Generation study was ask the community, would you want to know about things outside of Parkinson's genes? And a lot of people responded back and said, I only want to know about Parkinson's and the implication it has for my family members. And so today we've included the 7 key genes that are definitively linked to Parkinson's that have clinical actionability. So as we start to gather more research, we'll be able to also add to this panel and be able to update you with new information as we start to discover novel genes for Parkinson's. So this is an initiative that's already started. Dr. Shill is already enrolling participants into this study, so I encourage you to contact Barrow Neurological Institute. You can also participate at other centers of excellences through our Parkinson's Foundation network, as well as the Parkinson's study group sites. But given that all of you here today are mostly from Arizona, I highly recommend that you reach out to Barrow Neurological Institute so that you can sign up and schedule an appointment. Most importantly, given the COVID times, we are also offering telemedicine participation, which means that you can join an appointment by video call similar to Zoom like we are today, and not have to leave your home to be able to get access to the genetic testing. We also offer in-clinic based testing. So if you wanted to drive over to Phoenix to be able to get tested and do a blood draw, that's also an option. So I will just give you a brief overview of the 7 key Parkinson's related genes that are to the right here. Synuclein, SNCA, is also listed here. This was the talk that Dr. Shill had emphasized earlier. But I want to also emphasize the first 2 genes on this list, GBA and LRRK2. These are 2 really, really important genes linked to Parkinson's. Why? Because there are clinical trials currently ongoing and about to start that are looking for GBA mutation carriers or LRRK2 mutation carriers. I will also emphasize that there are other genetic tests out there like 23andMe that offer testing for GBA and LRRK2, but it's important to emphasize that this PD generation test is actually highly comprehensive. What that means is that we are taking the entire gene and detecting every possible mutation in that gene. So there could be hundreds of mutations in just the GBA gene alone. Now, in contrast, 23andMe is only testing 1 mutation within the GBA gene and 1 mutation within the LRRK2 gene. So you can see how limited the testing results might be for 23andMe. So we encourage everybody to consider getting testing through PD generation to get that comprehensive overview of your genetic mutation status. So key appointments to think about. If you want to participate from home, that's the online pathway on the left-hand side, and this is by telemedicine. It involves 3 key appointments. The first one is a very brief consent visit. So this is done online via video conferencing, and we are just doing a short visit so that we can collect some information about you, obtain your consent to participate in this research study, and then we'll be able to also get your mailing address so that we can ship you a buccal swab kit or a cheek swab kit to your home. About a week later, you'll have another visit, the main visit, and this is about a 2-hour-long visit that involves some clinical assessments, including a cognitive test and a motor test. At the end of that visit, you will also be collecting your cheek samples, and there will be 3 Q-tips in there, and then there will be a prepaid FedEx box so that you can drop that off at a nearby FedEx, and that will get sent back to our genetic testing lab in California. And then about a month later, you'll receive your test results through a genetic counseling appointment, and that would be done about 15 to 30 minutes, depending on your test results, through a phone call or by video conferencing. And this is an interesting component that's just been added because typically when you get your test results for any kind of test report, whether it's labs or other genetic tests, usually it's done by phone. That comes from your doctor. And in this instance, by offering video conferencing, it actually enables people to invite family members who might live outside of the state to also join this conversation. So we've had a lot of participants who have had even family members outside of the country join the genetic counseling visit so that they can also ask questions to the genetic counselor, which is a really nice feature. On the right-hand side, you'll see the in-person clinic option of participating. That involves obviously going physically to Barrow Neurological Institute, and that's about a 2-hour visit. And you'll go through the same clinical assessments, you'll sign the consent form, and then it will involve a blood draw. So that's the main difference, is that you would have to come in person and it would be a blood draw versus a cheek swab kit. And then finally, about a month later, you'll get your genetic test results through a genetic counseling visit. Again, this could be done by phone or by video conferencing, depending on your preference. So how to enroll? We encourage everybody to visit our website at parkinson.org/pdgeneration. There you'll see 3 key steps that you'll have to go through before you can contact a site to schedule your appointment. The first step is to watch our 7-minute PD generation video, and this just gives you another overview of what to expect when you come to your appointment, what type of testing this is, what the genetic test tells you and what it doesn't tell you. And then we also offer our consent form online. So that's step 2. You can download the informed consent so that you can review it ahead of your appointment. You don't need to sign it because you'll sign it in the presence during your appointment, in the presence of the healthcare practitioner there. So we will then lead you to this Enroll Now button, where you'll be able to contact an enrolling site near you. So you can again participate in person or by telemedicine. And if you're in Arizona, we really encourage you to reach out to Dr. Shill's team so that you can schedule an appointment for PD generation. So I just wanted to give you a live update of where we currently stand with PD generation. And despite our COVID pause back in March and April, we quickly pivoted the study to offer telemedicine participation. So pre-COVID, we were doing everything in person, in clinic, and COVID really pushed us to move into a telemedicine type of study. And that has done wonders for us because we have had enrollment from 33 different states. And at the time, we were only active at 6 different sites in 6 different states. So being able to offer telemedicine has expanded our reach across the US. And now people who live far from a clinic or might not have access to transportation were able to participate in receiving genetic testing. So we're very, very excited to be able to continue to expand. These are the different sites that you'll see on this map, and everywhere that's starred are areas where we will be coming soon. And just so that people know, we have over 800 participants so far in this study. Again, we're trying to enroll 15,000 people. So we do have quite a big target to reach, but we know we can reach it with your help. And PD Generation so far has been providing genetic testing for about a little over 100 people per month. So what to expect? Technology is going to be a major component to really think about. So if you're not tech savvy, which many of you probably are because you're already on Zoom joining us today, this is a place where you will require a computer and a webcam, and we may want to have family members involved to help you get set up. So again, either a family member or a loved one, care partner to help assist you and join that telemedicine visit. We do encourage that. The time that it takes to participate in this, again, it's 2 hours regardless of whether you're joining from home or joining in person. But just thinking about planning ahead, especially if you have to plan to take your medications so that you avoid off time, things like that, different snacks and staying hydrated, all of those things, just keeping in mind that you'll have to schedule in for that. And then the test results are provided through genetic counseling, which means that you won't be able to just get your test results unless you go through the genetic counseling appointment. So that is a requirement. And again, that's to really protect and ensure that you're getting accurate information explained to you live by a genetic counseling professional. So this is just a recap of how to participate. Again, parkinson.org/pdgeneration to learn more. Sarah has also put in the just quickly click on a link to be able to participate in the telemedicine study at Barrow Neurological Institute. And we also encourage you to reach out to us through genetics@parkinson.org if you have any questions or need help scheduling an appointment. Our goal is to be able to reach 1,500 participants by May of this year, and our overall goal is to be able to reach 15,000 people across the next 4 years. So with that said, I will pause there and stop sharing my screen so that we can then move into our Q&A session.
All right, great. Thank you, Anna. That was a fantastic presentation. We've received some great questions from our audience. So we're going to bring back both presenters and take some time to answer your questions. For those of you who haven't submitted your questions yet, feel free to do so right now. You can use the Q&A icon at the bottom of your screen. So let's go ahead and get started on questions. Anna, I'll have you go first. I think we get this question a lot. Which method is more effective when it comes to PD generation? Would you prefer a blood draw or a cheek swab?
So it's a really great question. And in fact, there's no difference in the quality of DNA. It's really the quantity that is different. So in a cheek swab, you get a lot less DNA than you would a blood draw. So obviously blood is king. But with that said, we don't want to limit people, especially in the COVID time situation. And so it doesn't matter really whichever one is fine, whichever one people feel more comfortable with. And again, the results are not compromised depending on either cheek swab versus blood. It's completely the same quality and the testing as well.
Excellent, thank you. Dr. Shill will give you this question. Anna covered the time commitment of PD generation, but what is the average length of other studies, or does it vary?
Oh, wow, it's a great question. So it varies tremendously. So I could give you a couple examples. I mean, some of our observational studies are just a one-time visit. You know, we're looking at some virtual reality goggles to be able to discriminate Parkinson's from other neurologic disorders, and that study takes about 45 minutes for somebody to participate in. You know, going along, if you look at we have a study going on looking at infusion therapies to manage on-off fluctuations in Parkinson's disease. That initial visit takes almost a day for people to participate, and then they're coming back, you know, once a week or every other week for quite a while until they get situated with the study medication. So some are quite lengthy. Some of our studies to slow progression go on over, you know, 1.5 years to up to 2 years in some cases. So they can be quite lengthy studies. So it really does depend on the study.
Yeah. Great, thank you. Okay, we had another question from our audience. Dr. Shill, I'll probably pass this one off to you. What are Lewy bodies exactly?
Really good question. As you saw, I didn't give you a lot of detail on that in my talk. So what we think what we think happens in Parkinson's at the kind of cellular level. So if you look at just the little neurons living in the brain, you've got these little machinery that's kind of taking care of things. They're taking care of producing enzymes to do things and proteins that live on the surface of cells that help us, you know, help the cells communicate, that type of thing. So that's being constantly turned over in the brain. And so, but what we think happens in Parkinson's is somehow that machinery sort of gets gummed up. And then you start actually accumulating some of these funny proteins like the alpha-synuclein protein. That's the kind of primary protein that seems to accumulate. So, and the Lewy body is the brain, the neuron's way, the little cell's way of trying to wall that off. So it just forms a little capsule around that abnormal protein and kind of walls it off to protect the neuron from the abnormal protein. So that's what we think. I don't think that's absolutely 100%. Known for certain, but I think that's kind of the basic thought about what the Lewy body is that the body's attempt to sort of protect itself from this abnormal accumulation of proteins.
Okay, excellent, thank you. As a follow-up to that, somebody said, I would like to understand the gut-brain connection of Lewy bodies. Can diet help? And if so, what changes are recommended?
Boy, fantastic question. The short answer is we don't know for sure. So there is a lot of interesting research going on about the gut brain connection. So we know that people with Parkinson's tend to be more constipated, and that's actually kind of part of Parkinson's, the neurologic control of the bowels. So we know the microbiome changes in Parkinson's. So what lives in our gut is changed in Parkinson's. So and there is some thought that that is relevant, meaning that the gut is actually communicating with the brain there. And there's some interesting study to say that actually might be happening. So, however, I think it's really too premature at this point to say, go change your diet and start ingesting this or start avoiding this in order to keep the Parkinson's from progressing. So I think that's still very much an area of hot research and kind of more to come on that.
Great, thank you so much. Anna, we have a lot of questions on genetics, so we'll jump over to you now. Once we know more about what causes PD in the end, how much do you think will be caused by genetics versus environmental factors?
What an excellent question. I think it's very clear that genetics has interactions with the environment. And so it's not just about genetics. Obviously, the global studies that are reported today indicate that about 10 to 15% of people with Parkinson's have a genetic form of the disease. Everybody else seems to have what we call sporadic Parkinson's, so no genetic form of the disease. So I think that's very interesting. And through PD generation so far, we've actually identified about 17% of the PD generation population carrying a genetic form of Parkinson's. So that's a lot higher than the reported information in the literature. That may or may not change, and it may go down as we start testing more people, but it does indicate that we are finding more people with genetic forms of the disease. And so there is an interaction between genetics and environment, and that balance is tipped depending on how much environmental exposure a person has had and the genetic composition of their background. So I think there are contributing factors, obviously, and so both are at play, and it's not just one or the other.
Thank you. So we had a person ask, can you discuss the SNCA mutation in more detail, and what do we do with a gene once we find out it is linked to PD?
So I can kind of start this one off first. The synuclein mutation is very rare. And one of the surprising things that we learned through PD generation was Dr. Shill had mentioned the PPMI study, which is a Michael J. Fox Foundation study that also looked for synuclein mutation carriers. And most of those individuals were in Italy or in Spain or in Greece. And interestingly, in the US, we did not expect to see very many synuclein mutation carriers. But within 3 months of launching this study, we found 5 synuclein duplication carriers, which was a very big surprise. All of them came from different areas, Chicago, Boston, and I think New York. And so what we're starting to see is that there is a prevalence of people with a synuclein mutation, even in the US, who are unrelated. And so it might be more prevalent than we thought it was. But more importantly, there are pharmaceutical companies that are working on drug targets around the synuclein gene. And so what we have been doing on the genetic side and through PD generation is partnering with pharmaceutical companies so that they can understand how prevalent these mutation carriers might be, and to be able to push them into studying this more and investing more resources into drug development for synuclein and other genetic mutation carriers. So we've been in talks with several drug companies now who have interests in pursuing this. And when it's so rare, it's difficult for drug companies to want to invest in that space. And so, money is a factor, but when they start to see that there are more and more mutation carriers of synuclein, they start to get interested. And so this is one of the reasons why PD generation is going to make a big impact, is to really de-risk the investment it takes for pharmaceutical companies to enter into Parkinson's to begin with, and then also for us to help accelerate their timelines so that they can get recruitment faster. The faster you you recruit, the more money you save. The more money you save, the less riskier it is to join into Parkinson's drug development. So this is all contributing, starting with the research, towards this ecosystem of getting more eyes on Parkinson's and more researchers involved.
Great, thank you. Anna, we have another one for you. How does the PD generation study differ from the one being conducted by the Michael J. Fox Foundation? And if I participate in this study, can the genetic results be shared with them?
Great question. So given that I'm an ex-Fox Foundation colleague, we work very closely with the Michael J. Fox Foundation initiative. So their initiative just launched a new version called PPMI 2.0. So they're also looking for mutation carriers. And what we are doing through PD generation is identifying people who might qualify for the Fox Foundation studies. And so anybody who comes through our doors who might qualify for a research study run by Fox, we're actually referring them right over, and they're also doing the same. So it's a really nice bridging of two foundations working hand in hand and sharing the research and the data back and forth. Importantly, I will say they are two different tests. And so the Fox Foundation, again, is only looking at specific mutations in certain genes. The PD generation test is a little bit more broad and more comprehensive in that we're looking for a lot more there. So it will help in terms of being able to supplement what you find through the Fox studies. So they are very complementary. We encourage everybody to do both.
Great. So say a participant participates in PDGene and they do not have a genetic link. Now what?
That's also a great question. So it's really important for us to, again, contribute the research data. And so just because today you may not have a mutation doesn't mean that you're free and clear, because we don't know enough about genetics in the Parkinson's space. And so there could be the possibility that we find more genes that are linked to Parkinson's disease. And so we are looking at everyone's data, whether you're a mutation carrier or a non-mutation carrier, on a real-time basis. So we've collaborated with about 40 global Parkinson's genetics specialists, and we've created an expert panel with the NIH that is led by the Parkinson's Foundation, so that we could start looking at the data in real time, so that anybody who doesn't have the genetic form of the disease, that's really important, because we want to see, well, is that really true, or are we just missing genes because we're not testing for them? So this is what's happening in the background in real time, is that we are looking at every person's genetic data so that we can detect more information and new genes that can come out.
Great, thank you, Anna. Lots of great information about PD generation. Dr. Shill, we have a couple of questions for you now. You mentioned certain biomarkers and biopsies. Can you further explain the role of research and diagnostics? And does having better diagnostics prevent PD, help people live better, or cure PD?
Right. So yeah, we were talking about a little bit about biomarkers. So a biomarker is a variety of things. I mean, it can be something that tells you whether you have the disease of interest, so you have Parkinson's or not. So it might inform you about how Parkinson's might progress over time. Or it might inform you about how you might respond to certain types of treatments. So there's a variety of ways you can use the term biomarkers. And then kind of, where do you get biomarkers? I mean, it can be something that you analyze in the blood or the spinal fluid. It can be a imaging type of test, or it can be something that you biopsy, where you actually look at tissues. So. And we look at all of that with Parkinson's. There's a variety of different ways we're trying to look at it. So some of it's in research and some of it's in clinical. So probably the best clinical example, besides just our clinical examination of diagnosing Parkinson's, would be something like the DAT scan. So the dopamine transporter imaging, which actually is kind of measuring dopamine levels in the brain. And so that's used to say you have somebody with a funny tremor and you're not sure is it Parkinson's or not. You can use that as a biomarker to say, yes, this person has dopamine deficiency and thereby has Parkinson's. So I think the kind of, in my mind, the more interesting things are some of these things like the skin biopsies. So because, potentially those could be used very, very early on, if somebody is just barely starting to show show some of the alpha-synuclein proteins starting to deposit, and so maybe a few years or no before they actually develop Parkinson's, you can recognize somebody as being predisposed to develop the classic symptoms of Parkinson's. So, and then the idea is that when we get these neuroprotective treatments, which we hope are coming soon, that we can intervene then as early as possible to try to prevent it from happening in the first place. So, and then even if people already have Parkinson's, confirming the diagnosis, I think you'd want to, especially if you're going to use something, maybe an immunotherapy or a stem cells or, something that might be kind of more aggressive therapy. I think you want to be really sure that, yes, this really looks like Parkinson's. And so if you can have something like that skin biopsy to really tell you that's what you're dealing with, I think that would be more comforting for us as clinicians to say, yes, this is the right population to go ahead and administer this treatment for. So a variety of ways biomarkers are used.
Okay, great, thank you. Another question for you. What about stem cell research in Parkinson's? You hear about it with other diseases. We used to hear about it a lot, and now we don't hear about it much at all.
Yeah, it's interesting. I mean, I've been in the business for, I don't know, more than 20 years now. And so I've seen stem cells kind of peak and then now kind of quiet for a while now. So, the problem, the kind of basic problem with stem cells is that you're trying to, what we used to think of Parkinson's as, just dopamine deficiency. If you could just put the stem cells right in that little dopamine area of the brain, you'd fix Parkinson's. And I think that was the initial report or approach to some of the stem cell studies. So, but now we realize that, maybe it's a little bit more than just dopamine deficiency. We talked about the loss of sense of smell and some of these different areas of the brain. And so, I think the idea of where do you put stem cells to actually have an impact. So, I think now that's a kind of problem, if you will, with stem cells is where do you put them? If you have to put them directly into the brain, what part of the brain is the best place to put them? So, and we know it probably doesn't work by just infusing stem cells. So if you just go get a blood draw with some stem cells infused in your vein, we know that it probably actually doesn't reach the areas of the brain that we want it to. So I think there's some problems with stem cells in that way. However, there's some interest in stem cells also from a kind of neuroprotective factor too. So, can you put these stem cells in to provide growth factors in the brain and actually just kind of give growth factors into the brain and sort of indirectly help Parkinson's. So I think that's still a potential area of research. So it's still going on. The research is still going on, but I think it still hasn't. We haven't figured out where it has its place in the treatment of Parkinson's as yet.
Excellent, thank you. All right, we have a participant who says, I was diagnosed with PD in 2019 and was told a cure was coming in 2 years. In your opinion, how close are we actually to a cure?
Yeah, so I guess that's a question for me, is it? So yeah, it's funny, even the Fox Foundation, they hope to have been out of business by, I think, 2010. So they hope to have had a cure within 10 years. And so unfortunately, they're still in business, but still actively working at it. So, how close are we to a cure? I've seen us come from research to try to slow progression, looking at, antioxidants and things like that, kind of these non-specific therapies that might slow progression of Parkinson's, to now much more refined, based on some of this genetic work that has been done, much more refined. We're actually really starting to understand the machinery that's going on behind Parkinson's, and that's leading to much more specific treatments. So I am much more encouraged these last few years about some of the things in development. So if I had to guess, if you think about a couple of the things that are, sort of in early phase development, it maybe is 3 or 4 years if some of these things really pan out. So crossing our fingers, that's the case.
I also just wanted to add to that, that, PD generation is a study where if PD generation fails and nobody wants to get genetic testing and nobody wants to share their research, their data for research, that will actually set us back about 10 years. So I think that's something that's really critical to think about, is how can we contribute our information so that we can really help accelerate these timelines forward. And this study will be one of those that's going to really contribute in a momentous way towards more effective therapies and also more drug companies coming into this space.
Great, thank you. Anna, we have another question about PD generation. Is it beneficial to do a PD generation study if you have no biological children, and would it still be useful for your own personal treatment?
Great question. In fact, we have Ann Hall, who is one of our patient advocates, who also doesn't have any biological children, but she's married to also somebody who has PD as well. And you know, for her, she's doing it for the research, but she also is hopeful that there would be customized treatments in the future that might be better for her, or maybe her dosage regimen might be more beneficial based on her genetics. And so there are things that people are looking at in terms of customized treatments, precision medicine. You might hear that a lot in the field, but based on your genetics, you know, you may have a better treatment course. And so it could help that individual themselves. And obviously the information is always valuable for the greater research good.
Great. And what would you recommend for those who do have biological children if they participate and find they do have a genetic marker?
Wonderful question. And in fact, that's the importance of the genetic counseling visit. It really depends on your children or your loved ones, whether they want to know about it. And this is a big ethical debate that's happening across other diseases. Alzheimer's is seeing this as well. And so, you know, first you have to figure out, would your children or loved ones want to know whether you have a genetic form of the disease? And that's actually part of the genetic counseling visit that does get covered. How do I talk about genetics with my family members? We provide a one-page how to share this information with family members, you know, one-pager as part of the genetic counseling session. We provide the actual test report. We provide a follow-up letter that is customized to that participant after the genetic counseling session to summarize the results and summarize that discussion, so that, you know, if you're ready and if your family members want to know, you can use that as a guide to help drive that conversation. But again, the genetic counseling visit enables you to invite loved ones into that conversation if they would like to be part of it. And I'll just share one anecdote. At Columbia University, we had someone who tested positive, and in the parking lot, she started calling all of her relatives saying that she has the mutation. And, you know, that was the one thing that it really is a preferential thing, but you have to think about the other person receiving that information and whether they would even want to know. So again, another interesting anecdote, but it's good to talk about that ahead of time with your family members.
Absolutely. Thank you, Anna. Dr. Shill, we have a question for people who may be interested in participating in research, but aren't quite ready to go out and about yet. So luckily with PD generation, we know we can do that at home. What about other research studies?
Yeah, it's a good good question. So, you know, we haven't, I'm not sure that we actually have the magic answer to that. So some of the studies do allow for virtual visits, so for people. But a lot of them, it's really hard to do the types of assessments. For instance, some of this, you know, the measures that we use to assess things have not even been validated for use over telemedicine. So, you know, for instance, if we have to feel the muscle stiffness or tap on a reflex or something, there's almost no way we can do that through telemedicine. So sometimes we really do just have to bring the person in for that. So. And so when we can, we try to do as much as we can virtually, but like I say, sometimes we have to bring people in. So, however, you know, we're bending over backwards to make sure visits are as safe as possible for people. So, screening everybody, including our clinic staff, and keeping everybody separated and minimizing the time that you have to be here for visits and that type of thing to just really make it as comfortable for people as possible. So we hope that with time that things get better for people and they do feel more comfortable. And so far, people have been pretty amenable to coming in for visits. So I think most people are pretty happy that they ended up participating. So. Oh,
good. Great. I think we are all excited to get out and about again. Dr. Shill, we'll wrap this up with a question for you. What is the difference between Lewy body dementia and PD? This person says they were first diagnosed with PD, now Lewy body dementia with Parkinson's. What does this mean for PD research if we don't always get the right diagnosis?
Great question. So really quickly, the difference between PD and dementia with Lewy bodies is really sort of what comes first. So with Parkinson's, typical Parkinson's, or what we call idiopathic Parkinson's disease, it's things like tremor and slowness of movement and shuffling of gait and those types of things as the kind of motor symptoms for Parkinson's. Whereas dementia with Lewy bodies, the initial symptom is a cognitive one, so memory loss or things along those lines. So it tends to start, I call it sort of a top-down Parkinson's. So those people can have cognitive complaints first, and then they might have some Parkinsonism later, or maybe milder symptoms of Parkinsonism at the time of diagnosis. So more cognitive or more motor, depending. Although I have to admit, there are some people that are kind of in between that you have a hard time knowing it, which thing do they fit into. So the interesting thing is, I mean, both of these conditions are conditions of Lewy bodies. So it's just kind of where the Lewy bodies are in the brain. If it's the dementia with Lewy bodies, they're more in the cortex or the thinking area of the brain. And if it's more the typical Parkinson's disease, then it's more in the brain stem where the dopamine neurons live. So there's sort of just two ends of the spectrum overall. So very similar conditions, just different and how they start.
Excellent. All right, thank you. With that concludes our Q&A session. Thank you both so much for taking the time to join us. This was a great way to engage the Parkinson's community in Arizona, hopefully get a few people participated in research, and maybe even a few people signed up for PD Generation. So thank you, Anna. Thank you, Holly. We appreciate you guys being with us here today. I do have a couple of more pieces of information if you guys don't mind staying with me for another moment, and then we will wrap up. Let me share my screen. All right, excellent. So if after everything you've heard today, you're interested in signing up for PD Generation in Arizona, here's a quick recap about how you can do that. If you're interested in participating in person, give Ruby a call at the Muhammad Ali Parkinson Center. You can see her phone number on the screen. If you would like to participate through telemedicine, you'll see a link in the chat. That'll get you through to a calendar where you can schedule your 30-minute screening call. And of course, if you have any questions about scheduling, feel free to give me a call, send me an email, and I would be more than happy to walk you through that process. And friendly reminder, we are here for you. If your question wasn't answered, I encourage you to contact our helpline at 1-800-4PD-INFO, where a Parkinson's specialist would be more than happy to hear from you. For more information about resources or upcoming virtual events, you can visit our website at parkinson.org, or for our local touch, visit parkinson.org/southwest. Last but not least, we hope you'll come back soon. Next up in PD Health at Home, we have Mindfulness Monday, where our expert speaker will lead you in a guided relaxation to help boost brain power and reduce stress. Then you can join us again on Wellness Wednesday for a unique and fun experience connecting with others through exploring art. That just about wraps us up. In the coming days, you'll be receiving a short survey, and we hope you'll take a minute to complete it. We'd love to get your feedback on this program and hear about other topics you'd like to learn about. On behalf of the Parkinson's Foundation, thank you again for joining us. Stay well and come back soon.